In March of 2020, when many of you were perhaps at Costco stocking up on Clorox and toilet paper, I was heading into the lab, anxious to run an experiment before the lab shutdown that we were all expecting the following week. I was about a year into my postdoctoral fellowship at a research institute in Seattle, and I was training in a leading lab that studied virus evolution. So as virologists, we were all well aware of the news reports that have been coming out from Asia and Europe. So in this week before we were going to get shut down, I had sourced a handful of reagents and was embarking on a pilot experiment that would tell me whether the experimental platform that I just finished building would have utility for understanding this new coronavirus that was advancing across the world. So how did I end up here? My initial interests in science came through the lens of evolutionary biology. Growing up, I was consumed by a need to understand why things are the way they are, and for me that search leads to evolutionary biology as the ultimate description of the origins for all of the endless forms most beautiful and most wonderful that surround us. I therefore embarked on undergraduate research with an evolutionary biologist at my college who studied the evolution of the Joshua tree of the American Southwest. At the same time that I was trudging across the Mojave Desert collecting samples, my other coursework was exposing me to a whole other world of endless forms most beautiful, that of the molecular world, the proteins, molecules, and machines that carry out our fundamental cellular processes and which themselves are products of evolution. I therefore specialized for my PhD in this field of molecular evolution, and through that ended up for my postdoc on viruses, some of the most concise and efficient molecular machines whose evolution plays out in real time every day. So here I was, pipetting away furiously, trying to see whether I could adapt my studies toward this new coronavirus called SARS-CoV-2. And I was interested in a particular part of this virus, these red proteins that stud the virus's surface, which are called the viral spike protein. Like a lock and a key, the virus uses the spike protein to recognize another molecule that decorates many of our own cells, called ACE2. This interaction unlocks entry into the cell for the virus, where it can then carry out subsequent steps of infection and spread. Because this interaction is so important, the viral spike is also a great therapeutic target. The dominant component of our protective response to SARS-CoV-2 vaccines, for example, comes from antibodies that we develop that bind to this viral spike protein to inhibit this interaction and thereby block infection. So here I was the week before the shutdown conducting this pilot experiment, and it worked. I proved that this experimental platform I'd been working on could query these different interactions of the SARS-CoV-2 spike, and that could help us answer an important question. If this virus can mutate and evolve over time, what does that mean for us? I think we all remember watching as the original strain of SARS-CoV-2 morphed over time into these Greek letter variants of concern, Alpha, Delta, Omicron, each of which sampled mutations in this viral spike protein that enhanced the virus's ability to infect us or evade our immunity. But how do we as a field keep up with this virus evolution? Traditionally, we've been reactive. We're really good at seeing mutations when they occur through genetic sequencing of clinical isolates around the world, but we don't typically know off the bat what that mutation might mean. And so it can take months to isolate a virus and see how it behaves in the lab, then see how it alters things like transmission or pathogenesis in animal models. We also have to understand how it evades vaccines and other types of therapeutics. We then have all these data that we have to integrate to sort of figure out what it all means. All the while, this variant has probably already swept across the globe, and the next variant is already cooking. So our approach was different. We figured, what if we could prospectively evaluate the impacts of mutations? If we could have a catalog of each of the possible evolutionary steps the virus could take and how that would alter some of its key properties, then the moment a new mutation is seen in a clinical sequencing data set, we'd already know what viral traits might be altered, which can guide public health recommendations. The challenge here is that there's a lot of possible mutations available. So there's 4,000 mutations in this viral protein, which exceeds the throughput of traditional methods for variant characterization. That's exactly what the experimental platform that I'd been building allows. High throughput measurement of the impacts of thousands of mutations on key protein properties. So here's what some of our data look like. What I'm showing you here is one of the sites in the viral spike protein, this site shown by a little star in the structure at the right. This site happens to encode for the amino acid asparagine, represented by this letter code N. There's actually 20 different amino acids, these different letters here, that a protein can use at a given site, each of which confer different biochemical properties. This means we can measure what happens if we change this asparagine to, say, a lysine or an arginine, which we now know would abolish this interaction with ACE2, as shown by the red coloring in the heat map, and so these are not mutations that are likely to evolve. Interestingly, though, this very same position can mutate to other amino acids that would actually strengthen the interaction of the viral spike with its ACE2 receptor, and these could represent an evolutionary opportunity for this virus. We have these types of measurements not just for this one position, but for each of the 200 sites in this viral protein, And we know how each of these mutations impacts not just ACE2 binding, but also how it allows the virus to escape from the antibodies that we all developed to protect ourselves against this infection. So we were ready. When this virus started evolving into new transmissible or immune-evasive variants, we already knew which underlying mutations were likely responsible for these adaptations and through what mechanisms. This work has therefore allowed us to contribute to efforts to track and forecast virus evolution throughout the pandemic. And we've also been able to use these experiments to contribute to the development of vaccines and antiviral candidates that are less susceptible to evolutionary escape by the virus. Now, while this work was ongoing, I was completing my postdoctoral fellowship, and so there were two important things I had to do for my own career. First, I had to think about the types of research questions I wanted to expand into as I transitioned into running my own lab. And second, I had to figure out where I was going to move to do that work. And that's what brought me to Utah. So I've been familiar with Utah for a long time. Growing up, the three staple vacations of my childhood were all road trips from our home in Minnesota to the Rockies, to camp in the summer, to ski in the winter, or to visit my grandma in Denver. I also got to know Utah in a more professional capacity during my research training. For example, coming out here in 2018 to a fantastic scientific meeting that was hosted by university faculty up at Deer Valley. Through these opportunities, I knew all about the great research happening here in virus evolution and virus biochemistry in labs like folks of Nels Elde, Wes Sundquist, and so many other labs across campus. So I was really excited to come here for an interview where I really got to experience the special research environment that's been cultivated here in Salt Lake City. I was really excited to get an offer and come here in 2022 to start my lab, moving here alongside my husband, Devin, who's a surgical resident here at the university hospitals. In terms of the research we're moving into, beyond studying the ways that a virus transforms itself during pandemic spread, we're zooming out to a broader perspective. Where did these viruses come from in the first place, and how can we stop them? This pertains to this concept of viral spillover, the process by which many of our modern-day human viruses emerged from broader pools of viruses that circulate in wild animal reservoirs. We have two main interests in this space. First, we want to understand the range of animal viruses that are capable of human spillover and understand how and why they evolved that capacity. And second, we want to develop vaccines and antibodies that can protect us against these diverse viruses that might be potential future threats. We were excited to really recently receive major NIH funding to embark on this line of research, and we're really grateful to taxpayers and NIH officials who support our work. Although this grant is going to move us into several different viral lineages, our initial work in this space has focused on a group called the SARS-related coronaviruses. So just as SARS-CoV-2 has been evolving in us over the last 6 years, these viruses have been evolving in bats for thousands of years, which has created massive evolutionary diversity, which we can represent on this structure similar to a family tree. Down here in blue is a cluster of viruses that includes SARS-CoV-2, but also a bunch of viruses from bats that are closely related to the human virus. In green is the cousin virus, SARS-CoV-1, responsible for an outbreak 20 years ago, and its close bat relatives. And then these other colors are yet other clusters of viruses circulating in bats across Asia, Africa, and Europe. Now, the field's conception of the sort of breadth of risk for human spillover on this tree has been pretty myopically focused on these blue and green clusters simply because that's where we've seen spillover occur before. Some of our recent experiments, leveraging the same platform I've been discussing, have shown that viruses from this purple cluster up here at the top of the tree, separated both evolutionarily and geographically from the rest of the family, are of higher spillover risk than previously assumed and resistant to many of the vaccines and therapeutics we've been developing for SARS-CoV-2. These are the types of observations that motivate our goal of contributing to pan-coronavirus vaccines and antibodies, reagents that can protect us against broad swaths of viral diversity so we can be protected even if we're unsure about which exact strain may cause a future outbreak. If we had tools like these for every single viral family, we'd be able to rapidly contain or prevent new spillovers, such that the events that we all lived through in 2020 can remain a truly once-in-a-lifetime experience. I'd like to end by noting that science is a team endeavor and a global pursuit. I'm grateful to the members of my lab here at Utah and my mentors and collaborators around the world who enrich the science I'm fortunate to be a part of. Thank you.